Datt. Datt has earned his PhD in structural and molecular biology from the University College London. He is now leading a research team at the University of Malaysia. His team focuses on structural bioinformatics, studying how mutations impact protein structures, with his research spanning host-virus interactions, antimicrobial resistance, and crop breeding. Today, Datt will be sharing his latest project, Democratizing Bioinformatics, Breaking the Bioinformatics Barrier in AMR Genome Analysis with AMRColab. This project is set to make advanced bioinformatics tools more accessible to researchers everywhere, especially in low resource settings. We welcome you, Datt. The floor is yours.
Thank you so much, Keegan. Before I share my slide, I want to take this opportunity to thank Ph for invitation. I think it's amazing to talk to all the communities and everything. So I'll start sharing my screen. Can you see it? And is it moving? Is the slide moving and stuff?
Yes, we can see your screen, Datt. Thanks.
So yeah, so good morning, good afternoon, good evening, everyone. I am Datt. I am from the National University of Malaysia, and yeah, I usually do structural bioinformatics, but this is a side project that we've been working on, and it's called AMRColab. If you heard about AMRColab, the AlphaFold stuff like that, you kind of have some kind of idea what is it. But for those that don't, you just stay tuned with us and stuff like that. Before we dive into AMR, I want to talk about this. So imagine this is like a thought experiment. So you just woke up from your bed, and then after that, you went to your bathroom, and then you open your medical cabinet. And then probably you just want to get an antibiotics from your medical cabinet. And then after that, surprisingly, when you actually open the cabinets, all of the medicine was gone. So there's no more antibiotics. And what you're left is just chewing gums. What? Yeah, so imagine this will be a nightmare. Imagine you just have a cut yesterday, or you just done a surgery yesterday, or maybe you are just have a scratch from a cat, and then all of these infections will get easily infected with all the like bacteria or microbes and stuff like that. So having no antibiotics or antimicrobial, it's a big project. It's a big problem for a lot of us. And this is not like a scenario that's not happening. This is something already happened to us. Like every day, you can hear people starting that, okay, there's already antimicrobial resistance happening in these drugs, another one. And this is it. And it is actually happening to us. But sadly, a lot of them are not really aware about it. But luckily, I think you guys are from the beach, so you kind of know what's AMR, I guess. So I'll go to my next slide. It's great to have the privilege to talk here because it's actually 2 days after this World AMR Awareness Week. I had a great time because I just went online and searched for a lot of videos. There are lots of posters sharing awareness about AMR. And it's super important for us to understand how important antibiotics are. And I went on a trip last year to Lahore, Pakistan, where at the time I was just invited to give a talk there. And then I was amazed by the scenery there. The people there was very friendly and stuff like that. And they talked my research. Do we have a chat? Oh, just okay. And then while I was there, and then it was amazing. The company was good. And then after that, the next day, I woke up with a running news. And then after that, I just went to the local drugstore. I thought that I can just get an antihistamines from them. But surprisingly, the people just gave me antibiotics. I was like, what? This is crazy. Like they give me a red pills, antibiotics, and they expect me to eat it. And for me, just we have the knowledge. We know that it's just a simple allergy. You just need antihistamines. You don't need such a strong thing to actually use it. So having awareness about AMR and also antibiotics could cut down the curve for the resistance and pave way for the drug discovery people to come up with the drugs in order to work in the futures and stuff like that. So in the year 2024, it's been amazing. We have heard stories saying that there's new antibiotics, zosularabulpin, and then the other one is cresomycin. Both of them seems to be having potential that can kill a lot of bacteria. Some recent stats, this is just 2 months ago, published in Lancet. So they're saying that antibiotic resistance infections could take up to more than 39 million people by 2015 unless action is taken. And then surprisingly, if you actually look at the map, this is what they predicted to have the highest death rate in 2050. You can see that countries is like South Africa, South Asia, South and Southeastern Asia, and also South Africa. And if you look at the map on the left, you can see the Argentina of the left on the bottom there, and even Russia and over some part of South America are actually getting some red dots there and stuff like that. So this is showing that it is kind of what is it called, serious now in these current locations, and that's why they're predicting for it to have a high death rate in these locations. This is worrying and stuff like that. And in 2017, World Bank actually did some calculation. So unmanaged AMR could shave up 3.8 of the global GDP per year by 2050 and bringing a lot of people to poverty. So don't worry, because I think there are lots of superheroes that's already there trying to help you and stuff like that, because I'm talking about all of this AMR surveillance team as all around the world, where I know probably some of you are actually from there, and you guys are doing so much work, where you guys are starting to collect sample from our environment, from hospitals, or even from human samples and stuff like that, in order to check how's the status of this AMR. And then with that data, people can actually do more things on it. So Phage is a company, is a society, is a organizations that cares about AMR a lot. And with all of those help, I think the government, in fact, the whole world, the WHO can come up with better guidelines and strategies to help with the AMR and stuff like that. And one way to really study them, usually when you have a sample, biologies, microbiologies, they will grow the sample, and then after that, they will do some what's they call chemical tests, and then see whether the bacteria are actually AMR. And then to really understand what happened to cause this bacteria to become AMR, whole genome sequencing is usually used. So what whole genome sequencing works is basically you just sequence the genome, get the DNA. With the DNA, you get the the genetic determinant of this organism, and then after that you can understand what are the AMR genes there, on in fact what are the mutations that are going to happen. This is going to help you to understand what happens to this microbe and how it's becoming resistant to a certain antimicrobial. So here's a typical WGS bioinformatics workflow. So usually the experimental biologists will extract the sample, and then after that the next part will be done by bioinformatic companies or bioinformatic expert. So once the company receives the sample, they will basically send the sample to the sequencing unit. With the sequence unit, they will run sequencing, and the magic thing that come out with it will be this ACTG. That's basically our DNA codes. And then after that, bioinformatician will start running bioinformatic analysis, predicting what's the AMR genes that are there, are there any mutations and stuff like that. And then they will come out with reports there. Surprisingly, usually we think that after doing that, and imagine you're experimental biologist, once you got the report, you think that, okay, the report have every single thing. But sometimes I do have experience recently. I just sent our data to for Meta's genomic analysis and stuff like that. When I got the result, I was so, I was so, what is it called? And I was so mad. Yeah. I was trying to tone it down. Basically, when I got the result, they just say that, oh, they run some basic pipeline, they don't know what is this and stuff like that. And when I'm actually asking more details of them, it's hard. So one thing that can be done, what if we can actually ask our experimental biologists to try to do some basic bioinformatic analysis and try to look at the result, then maybe you'll be better for their analysis to understand the whole genome sequencing. My work previously was on structural, but it was in this grant, basically Ph.D. basically give this subgrant where they obtain the main funding from Bills and Melinda Gates foundations, and then after that they provide subgrants in order to promote sustainable development in bioinformatics to support public health. So in one of the subgrants, they actually want to see the implementations of standardized bioinformatic practice, pipelines, and data structures in AMR in sequence laboratory labs in low middle income countries. So at that time, I was basically asked by Dr. Hui Ming here. She's actually from the teaching hospitals from the National University of Malaysia, where she's actually what's it called? AMR surveillance team, where every 5 years or two, she will do the surveillance and checks how is the series of the AMR happening in MRSA. And on the bottom one, this is actually the mentor of our whole team, Prof. Sheila Nathan. So she's actually a microbiologist, a retired professor. Yet this year she was actually awarded, she was actually nominated by the World of Academy of Science, and she's as a fellow in 2024. So under the influence by both of the group names and stuff like that. And you can see this nerdy guy, okay, that was me 2 years ago, trying to get from structural world, and now you're coming into genomics world and stuff like that. But we just learn from it. Apart from it, we bring in another person, Dr. Zanku Zeti Masdura. She's actually a medical doctor, and she's from a teaching unit from another university. This is called University Putra, Malaysia. So she served as our medical kind of like advisor on our team. We also have Dr. Mia Yang Ang. She just recently started his group in the Management and Science University in Malaysia. We also have a collaborator of Dr. Hui Ming. She's from Argentina, where she's currently working in the University of Buenos Aires as a researcher there. So this is basically our team. And the reason why I'm showing you these pictures is because this is the first meeting we have with Ph when we got the grant. So in this picture, you will see two important persons. So one is actually Emma Griffin, the other one is Finlay. Emma is actually the head of the data structures working group in Ph, and Finlay is a vice head according to the website. And then Jamie, I think, is some kind of admin person from Ph. So basically, this is the pictures when we first met, and then after that, Emma just briefed us what's the thing that they just have briefed us and what's the expectation they have for the project. So during the sharing, Emma did tell us after you have your genome sequences, usually what happens, bioinformaticians will run bioinformatic pipelines, and then after that, they will scan the sequences that you have of a genome towards a different type of database. And then after that, you will get different kind of reports depending on which of the software you have. So in different lab or different bioinformatics, they will have their preferred software. Maybe you'll use a software A, the other one will use software B, the other one will use software C. So when you're using different software, the naming of every single thing will be different. Even the name telling the gene is different. One is called OQXA, the other one is OQXA underscore, the other one is OQXA and stuff like that. So if you actually want to just collect all of these data and put it in some kind of like Excel sheet to analyze, this is hard. So Emma, this will actually cause the interpretation for it to be hard because it's time consuming. And then if you just by eyeballing using your eyes, looking at the report at the below, this is horrible. And you can't, human error may happen just from that. Luckily, Emma and Finlay's team, basically, they have developed this software called harmonization. Basically, what they've done in the simplest kind of way, they have harmonized the result from different AMR prediction software, where they have universalized the report from every single one, and then they put them in a simple format like below. So now you can say, okay, I'm using a software A, software B, and software C. All of the things are in the right place, even the naming of the genes are all correct and stuff like that. So currently, harmonization do allow more than 10 software. So most of the common ones are like the AMR Finder Plus, Ariba, REST Finders. So those that are using in the markets, you name it, they already have it in their package. So when we were asked by Emma to check how does this harmonization works and stuff like that, below are some of the insight we got from it. When we got it, we tested with some of a sample from Hui Min's lab. And surprisingly, when we run different software, we found every single thing was organized very well. And then it's very easy. And then after that, even running from different software, different database, everything was just organized very well. Even the header of the output was easy to interpret. And because of the success of the implementation, Ph decided to feature us in one of the blog posts. Thank you, Ph, for doing that. This is actually a motivation for our whole team, because for us, we are not really bioinformaticians that basically work on AMR that much, but getting some kind of recognition for AMR, we started thinking, can we do more, especially on this topic? And one problem when we actually found it, it's harmonizations needs to use Unix to run the software. So probably some of you will have questions. What's Unix if you're not bioinformaticians? So there are, I guess, 70% of person will have this question, because 70% of us are actually using Windows computer, where you have the Windows operating system. And then after that, there's another groups of operating systems called Unix. And then the most famous one are Linux and Mac OS. So if you actually just want to run harmonization on some of the codes, you need to use Linux or Mac OS to run it. But sometimes you can do some tweak on the window, but that will take some hours to set up. So imagine you're someone from the experimental side, you want to do bioinformatics, that will be very, very hard. So that was the problem. Apart from it, this one is just the machine kind of problem. Another problem will be for experimental people to actually start thinking about it. It's reading the codes, reading about the comments that you actually want to add. So for me, it's easy because I can read. Oh, the first part is installation, the second part is what and stuff like that. But imagine you're not someone that's been trained to read the codes. This sounds like jargon to you and stuff like that. And this is probably why a lot of people stop doing bioinformatics, and because this is hard and they don't like screen like this. It's just black or blue at the background and white wood and stuff like that. But yeah, that's happened in my life. And I'm facing with this almost every single day. So this is confusing, especially someone want to try to do some bioinformatics too. They can't because of the two problem and probably other problems as well. Then at that time, I was just thinking, what can we do? So I've been used, and luckily, before we got the grant, because I've been from the structural kind of group, there was a software called AlphaFold. This is software basically developed by the Google's company, where they have come up with a protein structure prediction software. The software is called AlphaFold. The lead scientists, John Jumper and Demis Hassabis, basically just got Nobel Prize this year. And this software basically can produce high quality 3D structures, high quality 3D models for a protein. And the accuracy can go up to 80% of them all the time. So making them on par on X-ray kind of X-ray crystallography structures and stuff like that. So that's the structures beautifully modeled by AlphaFold. However, running AlphaFold, you need crazy machine. Basically, you need to have a machine that have a GPU, and you also need to have a machine that have loads of memory. And sometimes this is hard for people from us that actually coming from a low-mid kind of country. So luckily, Sergey and also Martin developed Colab. So basically, they implement Colab, basically AlphaFold inside the Google Colab. And basically with that, everyone can start using AlphaFold to model the 3D structures that we have. Getting some inspiration from that, we started thinking, what can we do for the AMR world? So just to recap for those that are not knowing what's Google Colab, basically it's just you need to have a PC. It can be an iPad, it can be a Samsung tab, whatever tab that you have. You just need to have a browser, and then after that with internet access. Basically, just need to go to a website, and then after that, that website is the Google Colab website. And then after that, just clicking the connect button. After that, you'll be connecting to one of the computer inside Google's Google's company, and then you can use that machines for about 12 hours. This is good because most of the AMR genome analysis can be done within 12 hours and stuff like that. You don't need to set up anything. You just need to go to the website, and then this is free. So you get free access for like 12 hours, and Google Colab seems to fix the day. However, in order to implement the Google Colab there, me and my team started thinking, okay, what are the codes and how do we actually want to produce this Google Colab for them? So, and this is why we started thinking, can we develop a Google Colab for AMR Genome Analysis? So this is our initial idea, our prototype. Basically, if you have an access to a computing device, basically you just need to upload genome sequences of yours to the Google Colab, where we have adapted to now catered for AMR. And then after that, you can run bioinformatic analysis, where you predict the AMR genes and also variant with one of the software like AMR Finder Plus. And then after that, you can convert the AMR gene report to a standardized output using harmonization, meaning you harmonize your result. And then the only parameter you need to provide is just the file name. Is it a translated nucleotide sequence, or is it a protein sequence? Protein sequence with no genomics coordinate, and then just choose the organism you want. And then after that, voila, you get your result within a few minutes. And then after that, you can download your result after that in Excel format easily for you. And then you can start your interpretations and stuff like that. So this is the demo. Okay, I'm gonna stop it somewhere. So basically, this is one of, we have a lot of modules. This is just the AMR Finder Plus module. So when you actually come here, this is a special link that you just come, and then after that, you just need to come here and press the button connect. So this connect basically is the part that you connect to a Google computer. Just you need to wait. And then after that, doing the connect, after that, you get connected, meaning now you're connected to a Google computer now. And once you are there, you can run the recipe we have written for you to do your analysis, bioinformatic analysis. So this example is one AMR Finder Plus, where we're trying to predict AMR genes. So if you actually go back to before, we have instruction telling you how to install the software, how to test the software, upload the file, provide the genome sequences, parameters, and result. Okay, now we'll see them running. Next thing, once it's connected, you just press the play button. Then the installation for AMR Finder Plus will start. After that, testing whether the AMR Finder Plus actually works well. And then after that, I'm going to speed up. This is uploading the genome sequences. Then you can pick some of the parameters you have. And then just wait for a bit more, and you got your result. So all of this can be done in just one click through the computer. You just need to have your genome sequence with you. There's no need to write script. You just upload your sequence, and then you obtain your analysis within 5 minutes for just one genome and stuff like that. It's cool, isn't it? Yeah, so that was our module. So we do have another module that basically do harmonization. So similar, you will just connect, and then after that, installing, testing, running harmonizations, and get the result. So the reason why we built it to be another page, because we want to keep it modular. So sometimes when people just want to run harmonizations, you can just use the harmonization part. But if you want to use the AMR Finder Plus module, you can just use it. Okay. Once we got our result, we were happy because we did. And we actually want to share it to the public and stuff like that. So in 2022, we had our first workshop. You can see most of our team are actually there. So for this workshop, basically, we will start with some ice breaking. And then after that, we will talk about giving people what's the lectures on AML database, gene prediction tool, and then introduction to harmonizations. And after that, we'll break the room into 3 or 4 rooms. And then after that, we'll do the hands-on press sessions where we will demo the Colab with them. And then after that, we will ask some of them to present in the closing remarks to tell us how did they found about the software and stuff like that. In our first workshop, there's 32 participants. So most of them are actually staff or graduate students from local universities or from Malaysian laboratory in the Klang Valley. Basically, that's the near our capital site of Malaysia. About half of them are actually from an Institute for Medical Research. This is our basically the research wing under our ministry, where they focus on the AMR surveillance teams and stuff like that. And from what they from okay, yeah, so from what you can see here, before that you actually just need to do all those codings and stuff like that, but now you have this one on the right. And I was talking to one of the participants from the workshop, saying that this is easy and you don't need to worry about bioinformatics and everything. She was very skeptical of me initially and stuff like that. I was like, no, just trust it. And you just come to our sessions. I'm sure you can learn it and stuff like that. And surprisingly, after like the session, she came and talked to me and said that, oh yeah, it was so easy. I can't believe like science can be this easy. But I think it's because of all the hard work of all the people previously that makes the complicated science easy for everyone. And in fact, bioinformatics is not as hard if you have a right person to guide you on doing this. So we do a large scale a survey throughout the whole workshop, and then people find out they are more confident using our Colab. They are very happy with the harmonizations output, and then they are interested to participate in a more advanced kind of workshop. And apart from it, we also get some feedbacks from all of this workshop. There's some participants actually suggest us to add batch processing features. So if you're not a computation people, probably you don't know what is it. But let me just tell you. So imagine now you just have a machine. So this is, for example, this is your Google Colab and stuff. One of the module you have in AMRColab and stuff like that. So an original one, maybe you just give one sequence, and then after that, the Google, our Colab will process your data, and then after that, download the data, and then after that, you need to go back and upload your data, and then run it again, and you get your result and stuff like that. So this is very slow, and it relies on the people to come back and checking the tools and submit the data all the time. And with batch productions, basically now you just need to upload a zip file or WinRAR file that contains hundreds of like basically harmonized result. REST Finder result AMR predictions thing. And then after that, you just submit the WinZip file to the batch module, batch processing module that we have. And then after that, you can just process all of the hundreds data at one time. And then after that, you can just go and grab a coffee. And then after a while, you get your result for the harmonizations, and you harmonize your result instantly and stuff like that. So this is one feature we added in the next release. And then after that, even within our group, we're starting to talk about what can we improve with our module, with our Colab. So we're thinking, oh, can we add heat map? Because with heat map, you can now start to visualize all of the different sample you have, what are the genes that's available. You can see the resistance happening to a certain antibiotics. We can also add geospatial data, because we are probably interested, because imagine we're thinking now we have Sabrina from Argentina, from the other side of the world, and those in Malaysia. Can we just map all of this data online, and then we pinpoint it on the website, on the map, and see what's the changes that happens there? Apart from it, having a phylogenetic tree will be good, because that will tell the genetic relationship of the whole genome across our samples and stuff like that. So with that, we add one of the module, the visualization module there. So before that was 2, now become 5. And with this visualization module, currently it's not really a Colab that you can just run everything there. What we have done is basically just having some kind of instruction asking you to do every single thing, where you will basically generate your phylogenetic tree using pathogen watch. And then also you visualize your AMR surveillance data and a lot of metadata that you get from harmonization, and visualize it using microreact and stuff like that. And we're actually currently thinking, can we actually improve this module to make it basically less crazy, where you just need to run some code and just get it. You don't need to go to pathogen and microreact. But currently, it seems like it's hard. Yeah, I'm not gonna talk about the details there. Yeah, and now I want to show you the I want to show you the report that you get from this visualization module. Please bear with me because I know I just came out from this. Yep. So this is the result you get. When you have all of this, you will see, okay, this is the phylogenetic tree. So for us, we have 6 samples here. 4 samples are from Malaysia, 2 is actually from Argentina, and then you can see the phylogenetic tree of different sample. And then after that, on your right, this is the heat map. You can see these are all the genes, and then these are all the antimicrobial resistance. For those that actually blue means present, yellow means absent. And then you can actually zoom in and zoom out in your data set. If you have, this will be helpful. Imagine you have a lot of data there. And then after that, for the metadata there, depends what kind of data you have collected. If you have a lot of data, for example, you can have data from environment, from different lungs and stuff like that. This will help a lot of people like non-bioinformaticians to really analyze your data and get a full report like this and just populate it in your report that you need to submit to WHO or whatever after this. So this is basically the web page, and I'm going back to my slide. Okay, and then after that, we did our second workshop. This was done 1 year later. So we basically just talk about our phylogenetic tree and also the collapse. And this time around, we have a kind of similar kind of team. You have the icebreaking introductions and stuff like that. I still remember something that happened during this because in one of the virtual practical sessions, I was asked to demo one of the things. And then usually you hope that in this practical session, everything goes well. However, during that time, someone was just uploading some of the file where the file they have uploaded is ABCD space DEF dot whatever and stuff like that. However, because at that time it was actually a prototype Colab that we have, our module at that time doesn't allow you have a file name having this basically space there and stuff like that. And that our participant was just complaining the whole time and stuff like that. And then throughout the whole session, I was just nervous and stuff like that. But that's life. But I do want to thank that special participant for telling us sometimes people will have different naming, and it's good to standardize the name after someone have uploaded a file and stuff like that. And on the right, this is our group photos taken from it. So surprisingly, we got the same magic number, 32 participants. However, in this time, we got people from a more variety of microbiology fields. So someone from medical, food, environment, even metagenomic people as well. We also have people coming from outside the Klang Valley. So we have people coming out from Malaysia, the Borneo side, the Indonesia, Nepal, and even Hungary. So that's amazing because we're reaching a wider audience here. And this audience is actually more diverse. And we did more survey and we asked, okay, do they have any bioinformatics knowledge? And 70% of them doesn't really have a good knowledge of them. Yet we did receive feedback like this, saying that our workshop is helpful for non-biostatisticians, very informative, fun and easy to follow, etc., etc. Some statistics saying that they like the output, they are confident, and we recommend to people and stuff like that. And then after that, we did this post-workshop utilization survey 1 year after the workshop to see are they actually happy with them. And we found that 6 out of the 10 respondents have started using AMRColab in their work. And we're so happy with it because we didn't think that people actually started using that. This is good, yeah. And while we're actually preparing for our manuscript, we were actually expecting reviewer to be unhappy with us because of that. But this didn't happen. Most of our review were actually happy with our implementations, and one of the review actually started suggesting us to add a genome assembly module, meaning once you have done the genomic sequencing, there's assembly of the sequences to become what's it called? Of the data to become to assemble. Yep. And then after that, there's also another one asking us to add another AMR gene prediction tool, REST Finder, a widely used software. So we added this. So this is our current AMR version 1.0, where we have lots of module now. Currently, if you go to our website, no, not our website, our manual at the moment, we haven't updated. So we're in the process of updating the manual so that parts on the genome assembly module and also what's it called? The REST finder module will be there after that. But one thing to mention, our genome assembly module, it's still in the process, but depends on how big your genome you have. So imagine if your genome is very big, it's going to take a bit longer to run and stuff like that. But this happens even if you have a computer to run this genome assembly as well. So please take note if you actually want to run our genome assembly module. And then we published our paper recently, just came out last month. And then after that, if you want to read about backside story, behind the scenes story kind of thing, you can just read our blog post written by Dr. Hui Min. Probably some of you are just interested how's our future plan. So we do want to include more functionality in AMRColab. We want to allow for nanopore-based genome assembly module, quality control module, built-in phylogenetic module, metagenomic modules, etc. Improving the user experience, more comprehensive manual, video guides, translation to local languages, a website, and with a frequent update for people to get to know about our collab, and also on AMR news. And yeah, we're happy to do a workshop and stuff like that. So online and offline workshop, because this will be very helpful. We can collect feedback and usability and probably recruit people to train people to use AMRColab, AMRColab, yeah. And I'm sure some of you will think, this sounds great, but I'm not on bioinformatics and stuff like that. Yeah, in fact, Wei Min was just sharing with us, there was a professor, it's a colleague from her, were just saying that when people are talking about bioinformatics, this is talking like French students and stuff like that. And it's hard, but I don't think it's hard. It's just, if you actually look at what happens from our participants, lots of them don't really have a lot of knowledge on bioinformatics, and some of them are experimental biologists. If they can start to do some later process and starting to understand the data and everything, I think you can start to do it. So it depends what you actually want to do. You can basically talk to us and we don't mind to do a workshop for you, or we can come and talk to them if you are bioinformaticians, giving us some ideas how to improve it. We can work together on certain modules. Yeah, just talk to us, and we are very willing to collaborate with anyone that talk to us. Yep. So now's the acknowledgement side. We want to thank Ph for providing our initial funding, because without that initial funding, maybe we won't even got the inspirations to continue working on AMRColab. Thank you so much, Ph. And then after that, my team. So this is everyone. So Emma do join our team after we had the initial project, and then after that, she's been talking to us, giving us some advice on what to do on collabs as well. And our workshop participant, so far it's 64 at the moment, our manuscript reviewer, and also from the Ministry of Higher Education Malaysia. So this is part of another funding we recently got, where we're actually trying to do surveillance projects in Malaysia. And then this is just some pictures there. So if you look at this girl and the bottom pictures, the blue shirt one is actually Dr. Hui Min. Yeah, the AMR guru that started all of this thing. Last two slides. First, I want to let's end with a bit of imagination. Picture a world where AMR isn't a monster under the bed, but a problem we've conquered together. That's the world we're building with the tools like AMRColab. And I actually want to invite you guys to join the journey together. Let's get started and have some fun. And this is written by Dr. Hui Min. Our dream is that AMR will indeed help break the bioinformatics barriers in AMR research and surveillance, and allowing the research and public health community to always stay ahead of the AMR curve. And thank you guys for listening to the talk. And thank you, Ph, again. Thank you so much.
Excellent. Thank you very much, Datt, for that. That was an amazing presentation. I would now just like to open the floor for anybody who has questions. Please type them either in the chat or in the Q&A box at the button at the bottom. I see that we do have one question from Newton at the moment. They do say, great demos. Do you have this resource on GitHub?
No, we don't have it yet. And our plan is actually start with GitHub after this. Yeah. So we'll put everything in GitHub. Yeah. We actually saw that and we actually want to put it there. Yeah. So it will be there soon.
Excellent. I see there's another question from Anna in chat. Are there any projects to include pathogen identification?
Oh God, yeah, that would be very interesting. Currently, we haven't gone into that part yet, but this will be very, very interesting. Like, yeah, identifying the pathogens and stuff like that. Yeah. Because you do want to see something that's outside of the database or not just AMR genes. So, yeah, so definitely, yeah, that would be very interesting.
If nobody else has any at the moment, I do have 2 or 3 of my
Okay.
own, if that's all right.
Yeah.
So my first question would probably be, are there any concerns with regards to data privacy when using something such as Google Colab in order to perform your bioinformatics analyses? Because I do know that in the current age, there are many people who are advocating such a stage for the open access and standardization of data and data sharing. But at the same time, there have been cases of people misusing data that is shared with them. So I just wanted to sort of get your opinion and input on using a resource such as Colab in order to perform bioinformatics analyses.
Yep. So this is definitely something that we need to worry because especially you now have ChatGPT, everything you just type there, they will just learn from it. So sometimes people are advising me not to share some of the research idea there that at all and stuff like that. But luckily for Google Colab, what usually I will ask my workshop participants to do, they will connect to their Google Drive and then after that have the copy of that basically drive mounted and everything will be saved within their Google Drive. So it's everything within your own kind of like Google Drive. No one is going to have it. And what happens with Google Colab is after that 12-hour, or in fact, after 3 hours or 2 hours, after you disconnect from it, everything will be scraped away and stuff like that. So when you get to a new session, it's totally new, that all of the data will be gone. So the good thing is that's safe. But we did get some participants actually complaining, saying that every time I need to go there, I need to install it and stuff like that. Yep, we heard our concern. So in recent updates, we tried to make the installation taking less time. So some of it, we actually managed to cut it down to less than 1 minute and stuff like that. So yeah, so it's there. But make sure you save your data. But if it's mounted to your Google Drive, you don't need to worry. Probably it's there and stuff like that.
I see there's a question from Katie Hopkins. So for AMR gene identification, is it possible for the user to edit the thresholds for gene identification? For example, lower threshold for related genes.
Currently, we haven't added that yet because we scared that people can't really they want more simple kind of like what's that called menu and stuff interface. But yeah, thanks to Katie, I think we'll add that because it's quite easy. We can just add one line and then we'll allow people to lower that. But probably we'll add a few lines in the module saying that please be careful with it because yeah, lower the threshold may basically match to some of the genes that's not that correct and stuff like that. Yeah, so yeah, it's possible and we will do it. Thank you so much, Katie.
All right. If I may just ask another question so long.
Yeah.
With regards to using something like Google Colab, I know that they do to some extent provide free resources for people to use, especially with GPU, RAM, CPU powering and all of that, and they're quite high end. But what happens if somebody hits a threshold on the processing or the availability of it And sort of in a resource limited environment, what would the cost be expected for them to be able to sort of upgrade the facilities if that is possible?
Okay, so an easy way. So imagine now you use that computer and now you max out the limit and stuff like that, and you're eagerly to run it again. The easiest way is just to Google register another Gmail account, and then after that you get another 12-hour sessions to work on it, and then after that continue with it. That's cheating. I don't think that's good. But the other thing, if you're actually coming from a low-income kind of country, you may need to ask some extra fundings where you can actually go premium with the Google Colab kind of account. So those things will you will have access to a better kind of CPU, longer alarm time. You can even save your data there and stuff like that. So if you have the resources, go for premium. I forgot how much is the price now. I don't have it because yeah, even for me, I use Colab phones sometimes, but I usually install the software on a compute cluster and then we just run it and stuff like that. So I
Yeah.
haven't explored the premium part yet. So how much it is and stuff like that. Yeah.
Yeah. And then just sort of a follow-up. Somebody has asked, sorry, the sort of in comparison of using something like Google Colab to something like AWS, have you used AWS previously? I know a lot of people are also sort of shifting between the Colab versus AWS.
Yeah. So for me, I do know AWS. Basically, for those that are not familiar with it, basically this is some supercomputer with super clusters and connections. Can just pay some small amount of it, and then you can just go to AWS, and they do have some packages to help you to run on some genome analysis. They do have pipeline you can run and stuff like that, and you can have your space. Yeah, so they are totally helpful. But our target is actually for those that have low resources, and that's why
Okay.
we go for Google Colab and stuff like that. So you know there's something called Galaxy as well. So
Yeah.
Galaxy, yeah, there's a lot of software that's available. You can just pick whichever software that you have. What we see now is actually Colab is something to persuade people to start doing some bioinformatics too. And if you actually want to go for more high-end kind of thing, then you can go for Galaxy Web. But Galaxy Web, you need to take some time to really get familiar with. So a colleague of mine ran a Galaxy Web, and she found that, okay, if you just want to process 4 genome and stuff like that, you ended up having like on the right, you have some kind of outputs or runs and stuff like that. There are like 300s or 400s of lines that have been run. And sometimes you want to go back and track back which one is what. It's kind of hard for her and stuff like that. But there's some people do like the Galaxy part of well. So yeah, we see people starting with Google Colab, and when they are more confident going for the other one or investing in a Linux machine, or after that going for AWS and stuff like that. A slow process, one by one kind of thing. Yep.
All right, then we have some questions on the YouTube side, and then I see there's another one in the Zoom Q&A. So a question from Peter says that I can see that one can upload data using the browse button. I presume that the data lives in something like a Google Drive. So is there a way to push the input directory in some automated way? And then Arjun followed up on that saying they work on AMR Finder Plus, and with second Peter's statement, their team curates thresholds both for HMMs and BLAST, as well as the rule set to name genes. So
yep. So the first question is definitely possible to do that. One thing that we have recently asked in our recent Google Colab now, we are asking people to mount their Google Drive, especially you are using the batch version and also the genome assembly part. And we ask people to upload we teach them how to upload and using the browse sections on the left. But we do specify telling them which location and tell us where is the actual locations of the folder you have. And then after that, yeah, then they have every single thing. But maybe we should add another line telling them which is the part you should change, which is the source directory, and then that settle every single thing and stuff like that. And we can also think about another way of coding up. Yeah. Lots of ideas just bumping through my head and stuff like that. So what's the second question? Yeah.
Sorry, that was sort of a bit of a follow-up statement to Peter's question. There doesn't seem to actually be a question in that one, but there are two on the Zoom Q&A.
Okay.
The first one is, how does the pipeline deal with novel or new mutations? For example, a hit that is not 100% a novel mutation. Will it report the best hit, or will it have a special symbol to indicate that it is the best hit with a percentage identity to the best hit to date?
Okay.
And then the second question is, how often do you update the database?
Cool. I'll start with the second question first. So basically, the way we are extorting the AMR Finder Plus, the REST Finder, every single thing, we'll make sure we're using the recent website, recent version. So the one that they just released in GitHub, we will use that. And then after that, once you have using that, they will instantly use the newest database. So that's settled for question number 2. The first one, depends how novel is your genes and stuff like that. So if it's very, very novel, maybe that gene is not, it's a mutation, right? So currently, if you actually have a novel mutations, I don't think AMR Finder Plus is that bright at the moment. What they will tell you is maybe you get a drop of a sequence identity, maybe from 92.5 to 92.4 and stuff like that. That's also something that we can think about, isn't it? Like how to deal with it. Yeah. Currently, the thing is currently there's nothing that can do that, because we currently, how all of this software works is actually based on the database and stuff like that. But from bioinformatics side of the way, it's not that hard to do that, because if you deal with proteins, you kind of know how to do that. So I will think about how to do it and whether is it possible to implement that in our future. But currently we don't, the novel one won't be identified that well using our pipeline. In fact, by the AMR Finder Plus, as far as what my knowledge, that's what my knowledge. Yeah.
All right. Are there any other questions at the moment? Please feel free to ask. Cool. All right. I don't see any other questions. Then I think that I will bring this session to a close.
Okay.
Thank you, Mr. Datt, for a very amazing, wonderful presentation, very informative, and I really look forward to hopefully using the Colab book myself for any of my future research.
Yeah. Thank you so much, Keegan. Thank you, Phage, again. Yeah. Thank you, everyone. Yeah.
Thank you, Phage.
Yeah.